identify the key regulators of the glycogen and the glucose utilization systems by a simulation method. Oh, sorry. Maybe you can close it. Okay, sorry.
Doesn't need a microphone.
Okay. Actually, all this project is starting from a conversation between a biologist, Mori Sensei from NIST, and Matsuno Sensei, a computer scientist. They want to set up a computational model of a metabolome according to biological data. This biological data is the concentration map of the metabolome in E. coli, which include 100 more metabolites concentration in a time course. So our study, the first starting is from the starting point of the central metabolism pathway. The entry part is around the G6P, the first metabolite after glucose is uptake in. Then the second part, we want to extend our models to the central metabolism pathway, then extend to included other carbohydrate in, and then to all. Okay, in the previous work last year before, we have studied the starting point of the central metabolic pathway. In the previous work, actually we use quite a typical systems way, the cooperation between wet lab and the dry lab. Generally says is according to the experimental data we want to modeling, and during our modeling we found something different. So we do some predictions and then ask biologists to do experiment to test whether our prediction is right or not right. Fortunately, the experimental data confirmed our prediction is right. So after a comprehensive understanding, a new biological insight is found, and also a model is set. Okay, so our conclusion of the first part is glycogen is the primary source of glucose during the lag phase of the E. coli proliferation. This means in the lag phase, okay, the glycogen supplies sugars groups to G6P, and meanwhile, in the growth phase, excess glucose is stored in the glycogen for next proliferation use. Okay, according to the former studies, we tend to include the glycogen into the central metabolism pathway. Okay, now current our study is extended to the central metabolism pathway, the simulation here, the study here. Okay, since the model of metabolism, it can be looked at as a remote control toys, include the device, here is the metabolism, and the controller, here is the regulators, and the controlling method. Actually, these three questions is answer the question about what we do, and who control it, and how they control it. These are our starting strategies. And based on thousands of associated publications, literatures, okay, we filter out the related individual regulators of this glycogen and glucose system. Then after second selections, we connect their related regulators together. And then at last, we set up a circuit, regulation circuit, centered by HPR system, HPR and enzyme 2A. And this system controls the regulator mechanism, controls the metabolism of glucose and glycogen in this system. Okay, in this model, the device part included the PTS system, the glycolysis and the pentose phosphates pathway system, and glycogen metabolic system. Okay, the controller, the key regulators of this system we identified should include the HPR phosphorylation status system and HPR subcellular localization system. The phosphorylate state of HPR binding with GLGP will control the different glycogen decomposition speed. Meanwhile, active PTS, the enzymes of PTS is in unphosphated state, so it's associated with glucose and glycogen. Also, the HPR subcellular location, when there has a phosphate group from enzyme 1 transferred to HPR, so the HPR will move from the pole location, the head location, to the whole cytosol scattered. And if HPR is in the pole location, it's mainly binding with the GLGP work for glycogen. If scattered in the whole cytosol, majorly for uptake of glucose. Okay, and also including the gene expression controlling by the enzyme 2A and the CMP-CRP system, and also FDB-CRE system. I will describe this later. Okay, our regulation system can be divided in hierarchical maps, including the metabolism level, flux levels, protein levels, and the localization levels and the gene expression levels. Yes, and you see the blue one. The blue one is from PEP to enzyme 1 dimerization. This means that enzyme 1, the dimer speed of enzyme 1 is the bottleneck of the PTS system run. So this is the starting point of the phosphate group flux. Okay, comes here and controls two pathways. One is localization control. HPR is used for glycogen or for glucose. And also the phosphate status of HPR controls the speed of decomposition glycogen and also the glucose uptake speed. Okay, if detailed things, you can look at my paper. Since now, okay, the mechanism of metabolism part we have studied, and also regulator, we also know it. But the controlling method, we should, how we can start it, this one. Okay, we have to use the computational models to simulate. Then we can know how dynamically this regulation systems controls this device. Okay, so we have to construct our computational model. For the glycolysis models, the central metabolism pathways, the classic ODE models is from Chachanov, 2002 years. This is quite classic, based on 8. Okay, and existing PTS study, their models have been studied quite well by Rodriguez and Rowers. Yeah, their models is quite good and details based on the mass balance theoretical models. This name they called. Okay, but for the glycogen metabolism pathway, currently do not have specific to the special for glycogen, this metabolism pathway. I didn't find any computational models detailed describe this part. Okay, so we try to construct these models. In device part, we include the OD models from glycolysis, glycogen of metabolism pathway. This part I use a top to down method according to the concentration of both sides of metabolite concentration and to estimate the parameters. Okay, and the mass balance theoretical PTS modeling, and I will select all the regulation systems. Okay, the model 1 we set up in the Petronite. Okay, we transplant Chachanov's model of central metabolism into a Petronite model. Okay, successful. The result is similar. We got the same result as Chachanov's. The second model we set up is transplanting the Roa and Rodriguez model of PTS. Okay, result also showed similarity. Okay, then we integrate these two models to a combined one. The model 3 include the glycolysis and the PTS system. Then we add our selected HPR and enzyme 2A centered regulation system together with this model. we construct model 4. This is our final model, including that one, glycolysis and glycogene metabolism pathway and the PTS system, and also the HPR subcellular location control box and dimerized enzyme 1 dimerized PTS speed control box and another control box and gene expression control box. So run these models in Cell Illustrator, we got the result. And in this result, the red bar is the biological data, and the simulation result is the line. So the major metabolic concentration is a condensed. Only some didn't, not all the metabolic concentration we calculate the same to the biological one, but the major one is okay. And also include in that line, that rows is the PTS proteins behavior. And the end one, this line, the bottom line, is the regulator levels. Okay, how to analyze this result? We divided the whole cells, the E. coli life into six phases. Okay. In the first phases, you can see here, glucose and glycogen both have in E. coli. So this means that HPR is located in the head. So a little bit glucose active the PTS to be N-phosphate. So in the second part, the N-phosphate PTS of the pole located will catalyze glycogen in a very high speed. So this is the major position for glycogen is used. Okay, then glucose, not so quick glucose uptake has been activated because now the HPR has been activated, driven by the enzyme 1 to scatter from the pore area to the whole cytosol, work for glucose. So, but it's not so highly. So the glucose is uptake, but in a low speed in the later lag phase. Okay, come to the log phase, first part of log phase. Okay, this majorly glucose is uptake in this phase. We can see, yes, the sugar group flux speed is quite quick from glucose in this phase. Then from later log phase, different is now the extracellular glucose is less and less. So PTS that step by step to become phosphate. That means inactive. Okay, in the stationary phase, okay, no more glucose outside anymore. So you see all the HPR has shrinked into the head part. So in this period, the phosphate HPR catalyzes a low-speed glycogen for the E. coli to survive for a long time. If too quick, the E. coli will use up all the energies to die. Okay, logically say this is a composition of glycogen. We can see if from FDP to CRE system, we call this parameter alpha, and from phosphate enzyme 2A to CAMP-CRP system to control the GLGCA expression, we call the beta. Okay, this system's activation is like that. Okay, in mathematics, logical expression, I calculate it's alpha plus beta equals to gamma. Okay, this is a decomposition of the glycogen. Okay, the subcellular location of HPR is in the pore is lambda. Okay, if the HPR is in phosphate state, it's a rho, but in HPR in unphosphate state, nothing. Okay, and these both parameters controls data of decomposition of glycogen. Okay, this is a quite complex logical like that. Okay, now we can see in the lag phase. Okay, if